what it looks like under the microscope. This one is, I would say, pretty easy to answer without assistance. So this is just kind of like a work question. So 2 and 3 are the same thing. So what is the field of view under low power? So this is going to ask us to use the field of view formula. So what is the field of view under low power? What is the field of view under high power? And then it'll also want us to answer what is the field of view under scanning. Scan. This is low. And then high. They also ask you for oil immersion. We'll do it just so you guys can see the math, but that won't be on the test. So oil immersion, not on test. The other 3 are the ones we'll use on the test, okay? So when it's talking to you guys about the high power, low power thing, what it's trying to explain is the objectives. So one of these, need to use the matching colors. One of these, this is scanning, looks at the slide from far away. So it's actually not very magnified. It's only 4 times magnified. Kind of like if you're using your iPhone to take a picture, if you use like really close, you know, like you zoom in a lot, you're doing more times of magnification than if you zoom in less, right? So 10 times low is the next one. So that would be this one, this objective lens over here. And then 40x high, I believe, is blue. And so that one's over here. And then oil immersion is the last one, and we don't test you on that one. So depending on how much you're magnifying your specimen, the field of view will get smaller. Okay. And I'm going to go through all those right now. No, you do not have access to someone asked about calculators on the exam. So that's a good question. So you don't have access to any calculators when you guys are on your exam. Okay, so no calculators are allowed. And that's because we're not going to be requiring you guys to do a lot of work that would need a calculator. Okay, so don't like freak out that there's no calculators allowed. It's just because we teach you guys the right way to do your like your calculations to convert, right? We went over that last time, how to convert something from like meters to millimeters. So we go over that with you. And then for this part of it, like you guys are seeing, you don't have a lot of stuff on that. Okay, so let's see if it works for me to use my finger, and then if not, I'll just go over it on the computer while we charge it. So let's say you're using your 4x magnification, right? It's an objective. If you're using your 4x, your total magnification is going to be 40x. So when you plug that into your equation, 100x, right, times 1.83, this is our field of view formula, millimeters, over 40x, you're going to plug all of that in. So you can just take this x out because they kind of cancel. So 100 times 1.83 millimeters divided by 40 is going to get you 4.575 millimeters. Okay, so the field of view for low power, for scanning power, is 4.575 millimeters. You can memorize that. The field of view for low power, we're going to do the same thing, 100 times 1.83 millimeters over 100x, because 100x is the total magnification for low power is 100x. So when we plug that into our field of view formula, the answer we get is 1.83 millimeters. So basically the field, I'll write FOV, the field of view for low power is 1.83 millimeters. The field of view for scanning power is 4.575 millimeters. So you guys can memorize these, okay? So that's gonna cut out lots of time on your exam, and I'll show you what you do with the field of view formulas in just a second. And then the last two, right, the last two that you're asked to do in your packet is high. So the total magnification for high power is 40 times 10, right? That's how we get total magnification, which is 400. So we plug that in, 1.83 times 100 over 400, and we get 0.4575 millimeters. So this is the field of view for high power, 0.4575 millimeters. And we'll just do your oil immersion just so that you kind of have that answer in for your packet. So the total magnification of oil immersion is going to be 100, which is the objective, times 10, which is technically the ocular. So that's 1,000. So we plug that into our formula, 1.83 times 100 over 1,000. And that would be 1.83 divided by 10. Don't usually do this one, but I'm just giving you guys that answer. 0.183, right? So these would be the 4 answers to those 4 questions in your packet. Does that make sense? You would be, you want to write it out though. So you want to show your work. So you need to write this in your packet and the answer, not just the answer, okay? Because that part of the question that says show your work. So if you guys do that, those are the answers to the packet. Okay, so now I'm going to kind of try to show you how this works on the exam and what we use this for. Okay, so that would be what you'd be writing down on your packet. Let's say that you approach the microscope and you see underneath the microscope, that's what you see, right? It's like a circle. Field of view references the diameter of the circle. Okay, so the diameter of what you see, right? Diameter of what you see of that circle. So that's the field of view. So if you're looking at the field of view in scanning, I don't know why I just decided to do that. If you're looking at the field of view in scanning power, the field of view in scanning is going to be smaller, bigger, right? Because you're looking at it from further distance across. So scan is 4, oh dear. Oh, this thing is not 4.575. And we're gonna have to, did everybody get those? I'm gonna have to stop share because this thing is deciding not to work on me now. Okay, so those would be your answers to the next 4 in the packet. Okay, so I'm gonna put these on. Field of view. So again, that's the diameter of what you see. So for scanning, scan, which is the 4x, your field of view, we already did it, right? 1.83 times 100 over 40 equals 4.575 millimeters. So you're going to memorize that. Low 10x equals 1.83 millimeters times 100 over 100, because we multiply that 10x times 10, and then that gives us 1.83 millimeters. And then our pi, which is 40x, equals 1.83 millimeters times 100 over 400 equals 0.0 millimeters. So if you're looking at a question, and what we're going to do on the test is we're going to ask you to find the length and width of a paramecium. So if this is your microscope and here's your paramecium, okay, so let's say this is my little paramecium. They kind of have these little like fibers on them. Okay, so my paramecium looks like that. Now to find the length and width of that, you're kind of thinking, okay, well, how am I supposed to find the length and width of something that I can't? Sometimes I'll have a lot of students ask me, can I use a ruler to measure this? So they think, it's not a bad thought. It's like, could I take a ruler and try to measure it? So there's no way you can fit a ruler under a microscope, right? So that's why we use the field of view, because the field of view is the diameter of what you see. Okay, so the question is going to ask you, what is the length of the paramecium you see under the microscope? Okay, what is the length of the paramecium you see under the microscope? So what you're going to do is you're going to take the field of view, which, like we said, is this distance across the circle. That's the field of view. So if you know the distance across the circle, that really helps you measure, right? So let's say this is a low power 10x. Low power 10x. What is the field of view, the distance across the circle for low power 10x? What would that green line measurement be? Anyone want to take a stab at it? 1.83, right? So we already did the math for that. So good job. 1.83 millimeters is the distance across the circle. Okay, but we haven't answered the question, right? Because the question says, what is the length of the paramecium you see under the microscope? What is that paramecium length? So this is where you take a guess. You take a stab at it. So you will divide the field of view in use. So for example, this microscope is in low power, so we used 1.83 millimeters. Yes? What if the microscope had been in 4x scanning power? Wouldn't we have used a different field of view, right? So if the microscope had said 4x scanning, because you walk up to the microscope, you see that that microscope is in scanning power, you then know, okay, well, the field of view is changed now, because if it's in scanning power, my field of view is 4.575 millimeters. Does that make sense? So you're going to take a look at your microscope to determine the diameter of what you see. The circle inside the microscope is going to look the same every single time. What changes the technical circle is which objective is in use, because it gets you closer and closer to the specimen. Okay, so what we're going to do is we're going to divide the field of view in use by the number of paramecium that you think can fit across. Okay, so how many paramecium do you think can fit across the circle? So for this question, I'll kind of give us the answer, and then the next one we'll do it. So if we're looking at it, maybe we think that 1, 2.5. So that's 2.5. That's how many I think can fit. So I'm going to go with 2.5. So what is my field of view in use? We're going to say 1.83 millimeters. If I'm using 1.83 millimeters, I'm going to divide that by 2.5. 1.83 millimeters divided by 2.5. So that it gives me 0.74 millimeters. Okay, so the question asked me, what is the length of the paramecium you see under the microscope? The answer, 0.74 millimeters if it was in low power, right? If it was in scanning power, my math is still the same. I'm still dividing my field of view by how many I think can fit across, but this time, obviously, it's going to be a different answer. Divided by 2.5 is 1.83 millimeters. Okay, so usually students get really stressed out because you realize that you guys can't use a calculator to do this question. And it is kind of hard to do these questions like that. But usually on the test, we'll probably give you 2.5 to or 3, like a whole number or maybe a half. So it's not too complicated. And just another word of encouragement, this type of question, you're going to have 1 to 2 questions on this on the entire 50 question lab exam. So maybe 1 question, maybe 2 questions, but really just like probably 1 question. So it's worth it to learn how to do this. But at the same time, when it comes to this math, you're not going to have to do that a lot. Does that make sense to everybody? So don't stress about doing a lot of these. You might just need to do 1. So I would practice doing the long math, but it's not going to take up the whole exam because you only need to really do it 1 time. Okay. So the other questions that the test might ask you.
Sorry to interrupt, bro.
Yeah.
I'm so sorry. That's my son.
No, okay.
Yeah. So I have a question. You know, the number 5 and number 6 was saying approximate length, which is number 5. Then the other one is approximate width. How do you find out? How do you find for the length and for the width?
So the length we just did, right? What is the length of the paramecium you see under the microscope? That's the one we just did. So we're guessing how many go across the field of view. So you take the field of view and you divide it. So the answer that I have here is not the answer to your packet. I'm not going to give you all the answers to your packet. I'm teaching you how to do it so that you guys know when you go into the lab how to do that question.
Substituting the numbers.
Yes. So I'll try to show you guys another example just now. So if we look at this one that looks more like an actual microscope, we'll do another one here and then maybe that'll help see it 2 ways. So this one looks like a real microscope this time. So let's say that you walked up to this microscope on your test, or maybe you're using it in lab to fill out your packet. And in order to get the paramecium to look like this, it's on high power. Does that make sense? So everybody's looking at a microscope under high power right now. So how do you find the length of this paramecium you guys see? So how do you find that length? What is the length? And then we'll also do a width question in just a second. Paramecium. So first, you guys start with the field of view, right? So what's the field of view in high power? Anyone?
So it's going to be 0.4575.
Good. So 0.4575 is the field of view. What does that mean? That means the distance across the circle, no matter which way I draw it. So that could be this way. It would be 0.4575, that line I just drew. Or if I drew it this way, sorry, that's a little bit over. But if I drew it down the center, right? Because a circle measures the same any which way around the circle. So any way that I cut the pie, the way I cut it, right, is the same. So the field of view is any of these slices across is 0.4575. So now you just imagine that you basically sliced it right through the paramecium like this. And now you guess how many parameciums fit across that 0.4575. So 0.4575 millimeters divided by how many? So I think it's a guess. So you guys guess. So I think it looks like 2, and then most of 1, and then probably counting this, a third one. So I'm going to do 3. Does that make sense? Because it's kind of like almost 1 and then a little more. So 3 would fit across. Again, you are guessing this. On the test, your teacher and then like the lab assistant, they'll both look, Julian, and then if there's anybody else, they'll look at it. They'll all come to an agreement on how many they think can fit across so that the majority of the class is guessing that same thing, but there will be a margin of error. So like for this one, maybe they would do the correct answer could be if the student had guessed 2.5 or the student had guessed 3. Does that make sense? Because it's a guess, so it's not an exact thing, but you shouldn't be guessing 15. If you tell me 15 can fit across, I know you don't know how to do the problem. Does that make sense to everybody? 3 can fit across, 15 can't suddenly fit across the circle, right? You guys need to get kind of within a certain range. So let's do that math, 0.4575 divided by 3. So that's 0.1525 millimeters. Okay, so each paramecium length is 0.15, sorry, that's supposed to be a 5, 1525 millimeters is the length of the paramecium. Okay, so let's do a width question. So what is the width of the paramecium? If you're doing width, you're going to do the same idea, but you're just going to imagine drawing your line like a pancake through it. So like a stack of pancakes. So we're going to take the line, pretend it was this way. So pretend it was going through the width of your paramecium, right? Length of something is the longest part. Width of something is that shorter part across, right? So pretend you're drawing that line through the width. So now how many can fit across the circle width-wise? So how many paramecium do you think can fit from top to bottom of this circle, right? If I kind of pretended it was right here in the middle, how many could fit across? So 1, 2, 3, 4. Width is a lot harder to get exact because it's so big, so many. 6, 7. I'm going to say 8. Okay, so I'm going to go with 0.4575 millimeters divided by 8. Okay, 0.057. Okay, I kind of rounded it off. 0.057 millimeters would be the width of one paramecium.
But Brooke, under there they said the shortest portion. So what is that shortest portion? What does it mean?
That's the width. The width is the shortest portion because, like I just showed you, it's the narrowest part, right? So this part is the shortest or the narrowest part of the paramecium.
Another thing again is, okay, right here, you know, they didn't ask us if you're going to use the high power or we're using the scanning. So you just use the high power.
No, that depends on what you're using. For this example, I pretended we were using high power.
Okay.
But if you were using low power or scanning power, your field of view would be different, right? Because like we talked about, field of view for low is going to be 1.83 millimeters. Field of view for scanning is going to be 4.575 millimeters. So you need to see which of the powers which of the objective lenses is used to answer the question. So before you just start doing your math, you need to say, okay, which field of view is it? And then I can guess how many fit across and divide it. But you need to say to yourself which field of view is actually being used before you start just doing math. So for your test, the teacher is not going to tell you which field of view it is. You know, because you look at your microscope in front of you and you see which objective lens is being used. If it's a high power lens, which you guys need to know how to identify on your microscope, right, for the test. So if it's a high power lens, you use field of view for high power. If it's low power lens, you use field of view for low power and scanning field of view for scanning. Does that make sense? So you don't just start doing your math right away. You need to check which one is in use. Okay, so the last thing you should know is just to make sure you read your question. It is possible on the test that it will ask you to convert your answer into a different unit of measurement. You guys kind of, right, already have an understanding that you know how to do that. But like, let's say if it says, what is the length in micrometers? You need to know your answer is going to be in millimeters. So the answer we just found was 0.057 millimeters. You guys need to know, again, how to convert that to micrometers. We already talked about that last SI session. So if we go through the packet, we looked at our field of view. We looked at our 3 breakdowns of our field of view, right? And we solved those. So you would write those in and then find an example for this paramecium, right? You can do that in the lab. Or if you guys want, you can use the one in the packet to do the question. That's totally fine. So you guys can do this question 8, 9, and 10. You guys can do that at home using this picture in the packet. Okay, so you guys can use this to fill that out. All right, so for question 11, 12, and 13, and 14, we're going to use the starfish egg. So this is the starfish egg. And again, we can actually use this to do your packet at home. Okay, so you're going to draw and label a representative of the starfish egg. You guys can use this picture. You can just draw basically this picture and, like it asks, label the plasma membrane nucleus. So when you zoom in, you guys can see that there's actually like an outline here. So this would be the plasma membrane here, PM. This is the nucleus. This would be the nucleolus right here. And then this over here is the cytoplasm. Okay. So you can actually, you guys can label that right here. Okay, so then it asks us to find the length, the diameter of the starfish egg, right? So remember that for a circle, diameter is basically the same as length and width because a circle is the same distance across no matter which way we go. So if we're looking at, if we're looking at this example, let's say this example, let's use a new color. This example is in low power. This photo was taken in low power, okay? If this photo was taken in low power, can anybody tell me the field of view of this, basically this microscope we see? What's the field?
1.83.
1.83, right?
Yes.
We're going to take 1.83 millimeters and we're going to divide that by how many starfish we think can fit across the length. So why doesn't everybody, some people take a stab? How many do you think can fit across?
4.
4. So 1, 2, 3, 4. Looks like it might be a little more than 4, but 4 is fine. We can do 4. So 1, 2, 3, 4. We'll go with 4. Okay, so 4.83 divided by 4. .4575 millimeters. All right, so each starfish egg has a diameter, which is the distance across, right, of 4.575 millimeter. Remember, for the starfish egg, the diameter is the same no matter which way we go. Do you guys see how it's the same because it's a circle? So it doesn't matter. It's not like the paramecium. It doesn't have a length and a width. So basically a circle doesn't have a shortest part. It's the same every part. So you just kind of pretend which way we'll get it through the circle that makes the most sense, right? So 0.4575 millimeters is the diameter of the starfish egg. Okay, so just to be clear what we're about to do next, so I'm going to show you guys how to find, so this is going to be how to find the surface area and volume, and I'm just doing this for the sake of your homework. I'm going to write homework only. So the surface area and volume are not on the exam. Okay, so what is tested on the exam that we've gone over? What is tested? The parts of the microscope, the length and width of the paramecium, the field of views, right, of each objective. That would be scan, low, and high. And then the diameter of the starfish egg. So everything kind of we've gone over so far is all on the test. Good. But the surface area and volume is not on the test. So to find surface area, so I'm going to do it pretty quickly because I don't want to waste too much time on something that's just for your packet. But I'm hoping to help save you guys some time by showing you how to find the answer to it since it's in your packet. And you do need to complete it to get full points. Okay, so we found that the diameter of the starfish egg was 0.4575 millimeters, right? So we're going to find the radius next. So to find the radius, we take 0.4575 millimeters and we divide it by 2. The radius of something is always the diameter divided by 2. Okay, so we do that. So that's going to be our radius. Oops, 0.4575 divided by 2. 0.228. And I'm cutting off some stuff because this isn't an exact science here. So let's do, I'm not doing perfect math because this is a science class. So pi radius squared is our surface area formula. Okay, so I'm going to take that radius 0.228. And when you square something, that just means you multiply it by itself. So we're going to multiply that by 0.228. Remember, this is not on your test. We're just doing this for your packet, okay? And then we multiply that by 3.14, which is pi, 3.14, and that gives us the surface area of the circle. So 0.228 times 0.228 times 3.14 is 0.16, and then we're going to get the unit millimeters squared. That is the surface area of the circle. Okay, so 0.228 and 0.228 times 3.14. Okay, so this is the surface area. Okay, so now that we have the surface area, you're going to take your surface area and you're going to multiply your surface area times your, I think this is the way he's doing it, times 0.1 millimeters. The instructions are in the packet, so let me go to the packet. So anybody who's like a math nerd knows that this is not the proper way to find the volume of a sphere, but we're not really worried about that because again, it's just anatomy and this is for a question in your packet. So right here, assuming that one egg fits under the cover slip and that the thickness of the egg is 0.1 millimeters, which is what I put over here, what is the volume of the egg? And we're going to use the formula. So we're going to use the formula at the beginning of the packet right here. So the formula, volume of a circle, H times A, H is 0.101. He said 0.1, so we're going to leave it. I think this is the typo, this 0.01. And then A is pi radius squared. The two i's represents pi. Okay, so we're going to take our surface area that we already found, and then we're going to multiply that by 0.1 to find, where's my thing? To find the volume. So the volume equals 0.16 times 0.1, 0.016 millimeters, and then you're going to write cubed, millimeters cubed. Millimeters cubed, like this. Okay, so that would be your surface area and volume. Okay, if you want to use the example we did to put in your packet for the surface area and volume, that's fine. If you guys want to use the same example and put it in your packet, that's fine. But for the paramecium length and width, you guys need to know how to do that for your test. So make sure you guys practice that in lab by doing your own like work for that. Does that make sense? So pull out a slide, pull out a paramecium, practice the length and the width of the paramecium in lab. But for the surface area and volume, if you guys want to use the math I just did in your packet, that's fine. You can do that. Okay?
I have a question.
Any
real quick.
questions?
On the test, so you said there's like a margin for error, right? So like if they say like 4 can fit across and I say like 5 in my answer, as long as I like have the correct calculations, then they'll be like, okay, like that's fine. You just have to be like a close number.
Yes, as long as it's close. Like I said, if you're out here and you say 20 and I said 3, it's like, you know, but as long as it's close, that should be fine. There should be a margin of error on the thing. And you can always like, you know, you can always talk to Knudsen or Benders. So like, let's say you got a question wrong. You can always say, can I see, you know, the math that you got? And then a lot of times they have grace. Like if you got 4 and for some reason you were marked wrong and they had said 3.5, they can help you, give you back a point or something. I'm not saying that's guaranteed. I just don't want you to feel like super stressed about it. It should be fine if you were really close to the answer.
Okay, thank you.
Okay, 15, 16, 17, and 18. So this is our last question of the packet. And I know this has been kind of a lot to explain. Hopefully, though, it's going to save you time. If not, you're kind of getting the answers to most of the stuff as we go through it. So this is asking you about blood. So it's asking you about the diameter of the white blood cell and the red blood cell. And they basically almost have the same diameter. The white blood cell is a tiny bit bigger, but they're almost the same. And what we're going to use for that is we're going to use a blood smear. And you're not likely to be asked this on the exam because as you can see, these are really small compared to everything else we've done. So like I was just telling, I think it was Michaela that asked, when you guys are guessing, how are you supposed to accurately guess how many of this little cell fit across the circle? And me say, well, you know, I got 42 and you got 45, so you're wrong. Does that make sense? So we're really not going to be asking you red blood cell length and width on the test, red blood cell diameter. And that's just because it's actually very, very hard for you guys to accurately get that. So it'd be kind of unfair. In theory, though, you guys should have already heard, and you probably maybe don't remember because obviously this is still the beginning of the semester and there's a lot of information kind of coming in your ear and going out, but you guys would have already heard the answer to this. So in your cell lecture, and this is likely to be on your exam, in your cell lecture, he would have said the smallest cell, I think, yeah, the most abundant cell is the red blood cell, right? Do you guys remember hearing about this with your cytology lecture? And he said that the diameter of the red blood cell was what? 7 microns, right? And then he was talking to you about the oocyte being the largest, and that one being, I believe it's 120 microns. So that's something that could appear on your test. So take note of that, but we're going to do the math really quick anyway, just because you need to show your work. So you can't just write that as the answer.
Okay, Brooke, I have a question.
Yeah.
Okay, over here where you have estimate the volume of the egg using the provided formula on page 4. So the volume of the of a cycle is 4/3 times pi times r squared, which is radius squared.
So you don't use anything you find online. You only use what he gives you in the packet.
Yeah, that's what he gave us in the pack. He said on question 14, he said estimate the volume of the egg using the provided formula on page 4.
Yes.
Yeah, so the page 4, I actually went there. So the volume of a circle is 4/3 times pi times r squared.
So I just found it in the packet when I showed you guys. I'm pretty sure that it had been right there on right here. Right here, H, volume of a circle, H times A, H is 0.01, A is pi radius squared. So I took pi radius squared that I had already found and I multiplied it by 0.1 because this was a typo.
Okay, no, that's not what he gave us, actually. Is here is 4/3 times pi times r squared.
Oh, okay. Well,
Yes.
if he gave you a different formula, then you can use the formula he gave you.
Yeah, he gave us a different formula, yes.
Yeah, are you taking Bender or Knudsen?
I'm taking Bender.
So that might be a different formula because that would be the volume of probably a sphere versus the volume of a flat circle. So we're going, this volume is based off of a flat circle. A sphere has the volume you're talking about, and a sphere is like a 3-dimensional circle. So I think that Bender gave you you can just use that one, that's fine. So you can just do the math with that one. So you would take your pi radius squared that we found, and then you plug that into your formula. Does that make sense?
Thank you.
Yeah. Okay, so for this one, we're gonna take our distance across. So let's say that this person took this picture in scanning power. What would be the field of view that they use?
.575.
Good, 4.575. Okay, so let's just take a stab at it. How many do we think can fit across? If this is like, this is kind of how I do it in my head. I'm like, okay, if that's 10, something like 10, this is 20,
38.
40. I'm gonna say 60, but like I said, it's not gonna be on your test. We don't expect you guys to come to the same number, but I think we'll just say 60. Okay, so 4.575, 0.076 millimeters. Okay, that's the diameter of the red blood cell. And then if you were getting your diameter of your white blood cell, it is a little bit bigger. You guys can probably see they're close to the same size, but they're a little bigger. So I would probably say 50. So I would say 4.575 millimeters divided by 50. And then we're gonna get 0.09 millimeters. So that'd be the diameter of the white blood cell. Okay, so that's kind of, again, like your answer for those. Like I said, you're really not going to be, you're not going to be asked for those on the test in terms of specifically diameter of the red blood cell, diameter of the white blood cell. Okay, you do need to know the difference between red blood cells and white blood cells for the test, though. All right. Any questions on the microscope packet? So just to recap, what is being tested? You guys need to know the parts of the microscope, the length and the width of the paramecium. You need to know how to find that. If the only information you're giving is what power, how do you find the length and the width, right? We start with the field of view. We divide it by how many we think can fit across. You need to know the field of views of each objective, the diameter of the starfish egg, and then one more thing, you need to know the total magnification of each objective. So it is possible you will be asked, what is the total magnification of scanning power? Really easy, right? If you know scanning power is 4x, the total magnification is whatever your objective is times 10. So for scanning, the total magnification is 40. Okay, so again, what's our total magnification? For scan, it's gonna be 40 times. For low, the total magnification is 100 times. And for high, the total magnification is 400 times. Okay, so these are the things from the microscope packet that could be tested on the exam. Blood is something that can be tested, but that's also in your tissues packet, so I don't really think I need to rewrite it here. Any questions at all on the microscope packet? Hopefully that saves you guys some time. You can also use the math we did for the red blood cells. You're welcome to use that. You can use the picture, like on the packet, the picture that I just provided here. You're welcome to use that to fill in your packet. The only one that I would say you should probably additionally do in the lab is the paramecium, because students really struggle on that question on the test. So I would do 1 or 2 paramecium practice in the lab itself, but at home, you can practice labeling the microscope, maybe print out this picture or like on your device. You can practice, you can fill out your questions with this starfish egg and the blood practice questions. Okay, all of that you can complete from home. So almost all of the packet can be done from home. The last page of the packet has a labeled microscope for you. So if you're struggling to find any of the parts, it's all right there labeled for you. All right, any questions at all on this?
Yeah, I have a quick question. For the microscope, you said on the test, we have to remember all the parts, or you guys, there are going to be questions on all the parts for the microscope?
Like they'll be labeled for you? Is that what you're asking?
Oh no, on the test. So like, will they be asking us questions about the parts of the microscope?
Yes. And then there'll be this list of 16 are the ones you need to know.
Okay, so just the 16 or just everything on the microscope you recommend us or studying?
Oh, just the 16. We won't test you on anything except this list.
Oh, okay. Perfect. That was my question.
Does that make sense? So this
Yeah.
one right here has way more than what you need to know, but it also has what you need to know. So you can just pick out the stuff
Okay.
in there that you.
Okay, perfect.
Yeah, no problem. Like, I don't know, illuminator, the condenser you need to know, but you don't need to know the word illuminator. You know, recess
Okay.
for 45mm filter, no. But like the stuff on the 16 list, yeah, the 16 parts. Great question. I would say the parts of the microscope are very easy points because those are pretty easy to go over a few times and you kind of start to get them down, so.
Okay.
Okay, so let's move on to the mitosis packet. So I want to just remind you guys and encourage you, this first exam series is the first lab exam, has a lot of homework packets, but the next 2 exams, like the rest of the semester, there's only 1 per exam. Okay, so I know it feels like a lot right now, but it's kind of front loading your work. So for exam 2 and exam 3, you only have 1. So just kind of, you know, phew, good thing that you don't have all those packets later. Alyssa, you said Bender hasn't gotten your packets. You do have to do these. So if you're struggling to find them, you should be able to see them in your course. I'm just scrolling here for any Bender students.
Okay, because usually he prints them out for us to get in class.
Yes, and that you can still do that. Anybody here who, if you haven't had those packets, I know your professor can print them, but if you want to work on them or access them at home, you should be able to.
We can print them from home?
Yeah, you can print them from home.
Printing right now.
Because I'm the person who likes to do things that I can do ahead of time at home. So I would recommend just getting them done. And then that way, when you go into lab, you can be working on the things that you really need to work on in lab. Like, you know, like I said, look at the paramecium in lab. Don't be sitting there like answering a question that you could just go home and answer it at home, because your time in lab is so valuable. And when you guys get, so what I really want to like encourage you guys and really like spearhead you to is finish all the packets that you have this week, because next week you're going to be getting your tissues packet. And that is where like 60% of the lab exam will be about the tissues packet, because we're going to be testing you on different tissues. So one of the things that hurts students a lot is they feel so overwhelmed by the packets, like the mitosis, microscope, metric, that they get behind. And next week, when you guys get that tissues packet, you're like, oh, I'll work on that when I get time. I'll just finish up these packets first. And the big thing, you're really hurting yourself, because those packets, the little packets, are on the test. They are, meaning there's some stuff from them on the test. But the majority of the testing bulk is this tissues packet. So if you get ahead this weekend and you get all your packets mostly filled out, like 90%, maybe you have one more question on them. But if you mostly finish your metric, your microscope, and your mitosis packets, then next week you go into lab and you're pretty much prepared to start on that tissues right away. Does that make sense? So you're kind of keeping up, getting a little ahead so that you're not behind when that lab exam comes around.
I have a quick question. I was first on the waiting list for the first 2 weeks I'm in Newton's class, and I just got enrolled today. So I just bought the book today. I'm barely going to start with the lectures today. I have been in lab though, and I've done all the lab work. But as far as like the book and everything, lectures, I haven't done that. I'm going to get that started this weekend. Do you think that I will be able to have enough time to catch up for next week?
Absolutely.
As far as lectures, labs, everything.
Absolutely. I just will say that because I have tutoring today, and I can stay a little bit extra, I don't have to leave right at noon. But if you wouldn't mind, what would you stay after 11? I can take 5 minutes and kind of give you some pointers of like what you would want to do. And if there's anybody else in here who's maybe also joining into the class a little late, I'll go over it at the end. But towards the end, I'll just take 5 minutes, kind of give you some like, okay, this is what I would say to do with these areas of the class, and this is what I would focus on this weekend. But if you're just joining now, yes, you can catch up, but just don't leave right when we end, because I'll try to just give you guys some pointers on that, if that sounds good.
The only thing is I actually have to leave the SI in like a few minutes just because I have to go to work. So
gotcha. Well, I can try to message you, but I would just say, if I can summarize it quickly, do not need to read the textbook. Your tests are based off of the lectures, so you really want to spend time with your lectures and your note taking. Check out the SI right here. Where is it? My SI study tips and introduction to class. That's in the study resources module. Check that out, because you break down kind of how to study for the class. So I would really recommend looking at that, since I only have 2 minutes. So don't waste time on reading the book. Do your Connect homework quizzes. Just they're kind of like completion based. So complete those, and you don't need to worry about getting all the questions right. Make sure you study your lectures primarily. Check everything that's in the modules, and really focus on studying the lectures. So don't worry about reading the book. That would be my focus for you. So if you want to know what to do this weekend, go to the modules, do everything in the modules up to Monday's date, and watch all of your lectures and take all of your notes, and then that'll kind of get you on track for Monday.
Okay, and because my worry right now is the lab, right? So the first lab, I felt like it was pretty easy, at least for me. The second lab, what got me was I think I got it down pretty well. The only thing is that I didn't have enough time, right? So like right now I'm playing catch up on the second one. The third one, which I believe is this one, the mitosis, because I haven't seen any of the lectures, I have no really knowledge on anything in that. So I'm just a little concerned with that lab.
So this lab, and like I said, this is not the same way for the rest of the semester, but for the mitosis lab, nobody in here has gotten any of the lectures for it yet. So we actually do the lab, we do the lectures. And so what I would tell you is you can use this Google Drive link this weekend to work on reading it. The picture right here is the same as what it would look like under the microscope if you did the question, and the same picture. So I would, and this is what the teacher draws on the board, Newton draws on the board when he goes through it. So I would just use this handout and do your best to get ahead on the lab packets in the beginning of the class. The lab packets feel very overwhelming and they feel like the most important thing. But I will tell everybody in here that the area that you guys struggle on, that students struggle on every semester, is the lecture exams. So that's why I always tell you guys, study your lectures, study every word of your lectures, take great notes, and keep reviewing every single weekend. And then the other area that students struggle a lot on is tissue identification. And tissues is your next packet that's coming next week. And that's really where we'll be testing you the most, and you guys struggle the most. So in a sense, a student could not study mitosis, not study microscope, not study metric, and that's not the main part of the exam. Does that make sense? You're not going to get just to make it clear, you will not get a good grade if you don't study those packets. Please don't do that. But I just want to encourage you, don't waste your time like getting too far behind and fall back on like the part that really is going to make or break your grade, the tissues.
Okay, so as of this week, I'm pretty okay because we haven't started on the tissues yet. Since I'm playing catch up, should just study the lectures very well, don't read the book, do the quizzes online, and then finish up the packets.
Yes.
Okay, perfect. Because I'm kind of I feel like I'm barely getting started today, even though I was in the lab. So I'm like, I need to take this weekend to just get it all done by Monday.
No, you're good. I'm sure that was a great direction for everybody else in here for what you guys should be caught up on, right? Catch up on your lectures. Don't be behind on those at all. If anything, you want to try to be ahead. Catch up on all of your lab packets and get all of those filled out. So if you can do your lab packets and your lectures and be caught up and have notes done on your lectures and watch all your lectures and do your quizzes and do your packets, you will be in excellent shape for starting next week and feeling like you're actually gonna, kind of get that grade you want on the first exam, because that really is what sets you up for success in the class.
Okay, perfect. Thank you.
No, great. Thank you for asking. Okay, so for the mitosis packet, sorry, a few people asking questions in the chat. The tissue packet shouldn't have any color photos in it. Somebody, Alyssa, you were asking about pictures. You don't need to print the pictures at the end.
I didn't know. I was just trying to print them off. And so they printed off.
You're good. You
was
don't
like-
even need those. You just need pages 1 through 14. I don't know why the link was supposed to be deleted that even had those available.
I know.
So sorry that you wasted.
It's okay. Learn there.
Okay, so for the mitosis packet, really good information in the beginning. I would recommend reading through this intro part. Like we said, you guys have not had the mitosis lecture yet, but the mitosis packet has really good stuff in here. So you're going to read through all of this, and I will go over mitosis next week in SI, right? Monday we'll do the cell, and Thursday we'll do mitosis, okay? So question number 1 asks, what is the size of the daughter cells in relation to the parent cell? This is kind of a tricky question, and really, I don't think will appear on your test, but
Identical?
they're identical in genetic makeup, right? So in a sense, they're identical genetically, but actually it's kind of like humans, right? When we have a kid, that kid is definitely not the same size as the parent. And why? Don't they have like the same DNA? They do. They have the DNA, but they need time to grow. So when a cell replicates, it's actually the same thing. The cell DNA is the same, but the cytoplasm takes time to grow and completely grow up to size. So they're actually half the size of the parent cell in terms of like cytoplasm. The genetic size is the same, but the cytoplasm is half the size. So list several reasons for mitosis in plant or animal cells. These are actually given at the beginning of the packet. Mitosis is cellular division. It occurs for many reasons, with the most common being growth, repair, and maintenance. So growth, repair, and maintenance would be the list, several reasons there, okay? During which stage of mitosis is DNA replicated? Be careful here, think. So most of you guys will ask me and you'll be like, well, it's S phase, because you've read the packet and you see that it says S phase. S phase is correct, but S phase is not a phase of mitosis, right? So the true answer here would be not during mitosis, but S phase of interphase, right? So that would be the true answer. Growth, repair, and maintenance. Here. During which stage of the cell cycle are the centrioles duplicated? The centrioles are duplicated basically in two times. They're duplicated at the end of telophase in preparation for the new cell. And then they're also duplicated in G2 in preparation for cell division. And some of this is very confusing right now, again, because you guys haven't had the lecture yet. But once you guys get the lecture in about a week, it really does come together. So don't get too frustrated by the fact that, you know, you don't really get the answers yet. You're going to start to feel it come together. So basically the cell is making children, right? The cell is making copies of itself. Does that make sense? So one cell makes two daughter cells. And number one was same genetically, but half the size of the same size genetically, half size physically. So half the size is technically the answer. Okay, so for number 5, draw a cell cycle using G1, G2, S, and mitosis. So you guys are going to use this picture right here. This picture right here. Okay, so we're actually just going to copy that picture right into this box. So the picture looked like this. It was like 4, and it goes G1, S, G2, and then it has the 4 phases of mitosis. So that's P, M, A, T. So the definitions of each of these is G1 is normal metabolic activity of the cell. So the cell is just hanging out, just relaxing on the couch. S is when the DNA duplicates. So the cell makes a copy of its DNA. The cell copies its genetic DNA. G2, there is preparation for cellular division. So this is when basically all the products that the cell needs, it starts to get ready, okay? You can kind of think of it like the cell is getting ready for a big move or a big change and it wants to be prepared, okay? It has 2 copies of DNA. Now it's going to get ready to basically divide those and make 2 daughter cells. So then we have prophase, metaphase, anaphase, and telophase. And these 4 are the 4 phases of mitosis. Okay, so the 4 phases of mitosis. 1, 2, 3, 4. And maybe just quickly write this down. I would just recommend writing these down, guys, because this stuff will be on your lecture exam, and this having the basic definitions is going to really be helpful. So prophase, this is just going to, I'll just try to give the simplest definition. Basically, the DNA is coiled up, and then metaphase DNA lines up at the middle of the cell. So you can think metaphase middle. Anaphase DNA is pulled apart. And then telophase DNA separates into 2 daughter cells. So that's the simplest way to do that. And then basically each daughter cell then enters into G1. So each daughter cell would go through the same thing again. Does that make sense? They would go into G1, then S, then G2, then mitosis, each daughter cell. And then they would produce more daughter cells, and so on, and so on, and so on. Okay, so the original process is over here. And like we said, you guys haven't had a lecture. It's a little confusing right now, but that's just the basic kind of intro crash course, right?
The Connect chapter 2 that I finished last night covered some of this.
Oh, good.
Yeah, I was up till 3 in the morning.
I think I did. I watched
Because I like to
lecture
take
for that.
notes on everything, and I write down the questions, and when I go back to reading, I highlight the answers so that when I'm walking between classes or at lunch break or something, I could just be reading my notebook.
Good. And hopefully when you get that lecture too, that'll clear up a lot for you. Okay, so what is the name of the stage between cell divisions called? What is the collective term for the stage when the cell is not undergoing mitosis? So G1, S, and G2, right? That's when the cell is not going through mitosis, and that has a name, and probably somebody is already either putting it in the chat or definitely knows what I'm saying, interphase, right? So all three of those are interphase. So mitosis is prophase, metaphase, anaphase, and telophase. Interphase is G1, S, and G2. Okay. So now let's get down to kind of the nitty-gritty part of this packet. So for what is on the test. So this is the part we're doing now that'll be on the test. Identify the 4 phases of mitosis under a microscope. Okay, identify the 4 phases of mitosis under a microscope. On the lecture exam, questions about mitosis. So for everything we did so far, questions 1 through 7, that was all stuff on the lecture exam. Does that make sense? It's not really going to be on your lab exam asking you all the questions about mitosis. It was just trying to help you guys get a basic understanding before you did your packet. Not on either exam is calculating percent. So this part right here, I'm going to show you guys how to do number 8, and it'll be number 10 down at the bottom, but those are not on the exam. Does that make sense? We're not going to be calculating percent on the exam, but I'll show you how for your packet. So the main part of the exam is identifying the 4 phases of mitosis under a microscope. So I'm going to start with that because I feel like it's nice to really start with the thing on the test, and then we'll go through what's on the packet. Okay, so here we go. Here is an onion root tip. So this question right here asks you to obtain a slide of onion root tip. So when we pull up, I have it labeled allium root tip. So you're going to open that, and this allows you to complete the packet at home. So if you guys wanted, if you guys wanted to do this weekend, you're welcome to using this packet. Okay. And obviously right now I'm also going through it with you, so we're kind of doing it together. So we're going to do what's on the test. Identify the 4 phases of mitosis. That's our goal right now. So we're going to look at prophase first, right? Because we have those 4 phases, prophase first. So in prophase, the DNA is coiled up. So this one is in prophase, the ones I'm outlining. This one's in prophase, the DNA is all coiled up.
Yeah, let's go. I don't know.
Devine, do you have a question? I'm not sure if you had a question. I just, I heard your mic was on.
No, I don't. Sorry about that.
No, you're good. You're good. Okay, so there's another prophase. And okay, so those would be the 3 that I would say prophase. If you guys are talking about your exam, we're going to make it very clear. So the best example right here is this one. Okay, so this is the best example of prophase. I kind of did these 2 because they're also good, but truthfully, the one that we would do on the exam would be something that looked like this. Does that make sense? Because that is a best example of prophase. So we're not going to give you weird ones. So prophase, I think it looks like scrambled eggs. Does that make sense? Kind of like all scrambled up, coiled up DNA right there. So then what did we say happens in metaphase? We'll use a new color. So in metaphase, we said DNA lines up in the middle, right? At the middle of the cell. So let's find one that looks like that. So this is a metaphase. The DNA is kind of pushing itself towards the middle. Do you see that right here? How the DNA is pushing itself towards the middle. Again, another example here of metaphase with the DNA kind of pushing towards the middle. Okay. And then let's see. So those are the two best examples there of metaphase. So metaphase, I just think of the word middle. They're kind of pushing themselves towards the middle. All right, anaphase, let's use red. So anaphase right here, this is a great example of anaphase. In anaphase, they are pulling apart. Anaphase, pulling apart. Do you guys see that? How they look like they're pulling apart from each other? Yes? Kind of like they're starting to detach on one end, right? But they're kind of like still attached, but they're pulling apart, right? For this one here. Yes? Okay, so then telophase and telophase. This is the best example of telophase. Okay, telophase kind of looks like they are coiling up at two ends. So two coiled up balls at either end. Okay, so 1 and 1. 1 and 1. Okay, so those would be our best examples of each of those phases. So what's on the test? You guys need to identify each of the phases of mitosis on the test. Any questions or clarifications on that? Usually somebody asks, what about interphase? So every other cell you see is in interphase. So if I didn't draw a box around it, it's in interphase. There are some of the ones on there that really look probably like they're supposed to be in like prophase, metaphase, anaphase, telophase, but I didn't draw a box around them because they're not clear examples. And like we said, if it's on the exam, you guys are going to get a clear example. So I didn't want to put things that would confuse you. So we can say every other cell you see besides the one that I drew boxes around, those are all in interphase. Good. So right away, can we all agree that like 99% of the cells are probably in interphase, right? And not as many are in prophase, metaphase, anaphase, and telophase. Yes? Okay. So what we're going to do is we're going to really quick, we're going to look at the other slide for the phases of mitosis, and then we're going to do your percentages calculations for your packet. And then once we're done with that, you'll basically be done with math for the semester. Okay, so after this exam, math, you're leaving math behind you. So I know it feels like a crash course in math. You didn't really expect that from anatomy, but there you go. Nursing school is a lot more math than you would think. There's a lot of dosage calculations and medication calculations. So it is good to keep your math brain a little fresh for your dosage calculations and medical calculations when you get to, if you're doing nursing.
I have a question really quick about the lab exam.
Yeah.
Is it going to be like on like basically like a lab packet that we have, like where we write in all of our answers, or is it like on a Scantron?
Scantron, and it should be, I kind of had gone over it Monday. It's going to be multiple choice with none of these as one of the choices. So you just kind of need to prepare mentally for it to be a fill in the blank. A lot of students hear multiple choice and they kind of think, okay, well, that means it'll be easier because the answer will be right in front of me. But actually we use none of these choices like 50% of the time. So it's kind of a 50-50 of whether you see A, B, C, or D being correct, or whether you need to know your information well enough to say, okay, well, the answer is actually not right there. It's actually none of these choices is right because it's a different answer. Does that make sense?
Yeah, I do remember you saying that. I wasn't sure if it was just the lecture exam that was multiple choice that way, or if it was the lab also.
Got it. Yeah, no, there was an overwhelming amount of information. I know this class is like a lot to get the hang of at first, but yeah, so multiple choice. There might be a few fill in the blank. I don't want to 100% guarantee all multiple choice, but it is taken with a Scantron, so that, yeah, like we said, so that it's just cut down on grading time too, so you guys get your grades back a lot faster because they don't have to be manually graded. And then for like your math, he should be allowing you to bring like a blank piece of white paper. So you should be able to ask him, can I have a blank piece of white paper to do your math? And then if not, the back of the Scantron has a lot of places to, like, it's all lined, so you could do your math on the back.
Thank you.
Yeah, for like your metric conversions and then for the microscope paramecium. Okay, so this is the whitefish blastula. This is not an excellent photo, but there are some examples on here. Typically, I'll just be honest with you, typically we use the onion root tip that I just showed you because most of our whitefish blastula slides are really not excellent. But at the same time, you could get a whitefish blastula on the test. So I'm going to try to show you guys more clearly what each one would be. So this here is anaphase. Sorry, I should try to go in order. Prophase would be, this one is kind of like a prophase. Like I said, these are not as good quality, these slides, so they're just harder to work with. That would be anaphase. This would definitely be a metaphase. You guys can see it's at the middle. This one is basically a metaphase right here at the middle. Anaphase, you can see they're next to each other pulling apart. And then let's use a different color. Telophase right here, because they're starting to kind of coil up into two balls at either end. And this one I would say is a telophase. You can kind of see in this example, do you guys see how this telophase, it's kind of hard to tell, is that telophase or is it anaphase? Because we can still kind of see the spindle fibers, the little like lines pulling apart. And what I, again, will just remind you is the test, we're not trying to trick you. The example we give you, okay? So the example we give you will be clear. The example we give you is not going to be super tricky like this. So don't really stress about that. We're going to give you a clear example of one or the other. And 9 times out of 10, we use the onion root tip, and the onion root tip is a lot easier to see, but the whitefish could be on there. So I don't want to 100% promise you that. It has been on tests in the past, but usually we use the onion. Yes, good observation, Alyssa. So the anaphase is kind of fluffier. Good.
Yeah, I just put that in my little notes. Fluffy
like
presents.
Okay, so let me try to break down the percentage thing for the packet as fast as I can. Again, it's not on the test. So I'm going to try to do it really simply, really clearly, so you guys can just do that, kind of get it out of the way. And just reminding you what we're doing. We're looking at this part where it asks you how many number of cells and how many percent are in that phase. So I'm going to try to do that as fast as I can. Just bear with me because it's a little bit of a process, but I'll try to make it really simple. Just write some steps and then you guys can get it down. The last question of the packet, just so that we finish with percent. We're not finishing, by the way, because we still have 20 minutes. And I'd like to go over some, I think I'm going to do, maybe I can do a poll. I'll probably do models. And then I'll just, for chapter 1, I'll just post my outline of my notes. And we'll just kind of skip doing chapter 1 in SI, and I'll post an outline of my notes. And then on Monday, we'll do chapter 2 in SI. I think that would probably work. So I think we're going to end with models today. So I think I'm going to do the mitosis models and then the cell model today before we go. And like I said, I'll post an outline of my notes on the announcements.
A question real quick. The outline, is that going to be like your, oh, what did you call it on Tuesday? Concept map. Is that gonna be like
Yes.
a content round? Okay.
Yes, and I'll probably post a few because like I'm gonna have one for the movements because the movements in and of itself is kind of like a separate thing. And then I should have one for the other thing. Because chapter 1 is so short, if you guys want, if you guys are interested, I might just post all of my notes for chapter 1. I only have like 8 pages of notes. And then you guys can kind of see how I did chapter 1 when I was in the class. And I'm only doing that because chapter 1 is very simple and I want to show you guys exactly what you need to know for the test for chapter 1. And then that'll kind of help you with gauging how much time you're spending like outside of the lecture. So I just want to make sure you guys are not wasting your time going into too much detail on things that you really don't need. And again, Monday, chapter 2, we're going to go over that all together because that is a really kind of in-depth lecture. But for chapter 1, being less in-depth, I think our time is better spent on the packets, if that's good with everyone. So I'll probably just post, I might just post all of my notes for chapter 1 so you guys can look through those, get a good feel, see it all, and then I'll post the outlines too. I think that probably would that be helpful? That would probably be helpful. Okay, so for question number 12, asking you to write the order of animal cells. You're gonna use these right here. Let me erase like all of this writing. Okay, so you're gonna use the order, the correct order, and then you're gonna use these pictures. So the one that is pretty obvious, I'm trying to scroll out here. The one that's pretty obvious, that is the first one here, is this one, right? This would be pretty, in my opinion, it's pretty obvious that this is G1, right? The cell looks like it's just resting, not doing anything. This is S phase, right? The cell is now duplicating its DNA and preparing to divide. You could either say this is S or G2. Either way would be fine. Because really we're just numbering them. So for example, I'm doing 4. I think that that's the first one, and that is right. 3 would be next. Yes? Okay, and then now the cell is going to enter into prophase. So it's going to kind of scramble itself up right here. 6. Then the cell is going to line the DNA up at the middle, right? Right here in metaphase. So that'd be 5. Then it's going to start pulling that DNA apart, but it's still in metaphase. It's just barely, well, this would be early anaphase because it's just starting to pull apart, but it doesn't really matter. 7, because that would come next. Does that make sense? This is really technically, sorry, I'll erase this. Technically, this is anaphase because they've already started to pull apart. So I'll write A. This would be later in anaphase because as you can see, they're really pulling apart in 1. Okay. And then anybody, we don't have a lot of options. So do you guys think that number 8 comes next or number 2 comes next? 8 or 2?
8.
8, right? So we're going to do 8 next. And then 2 would be last. Good. So when you look at your packet here, oh, wrong one. You're going to write this, right? 4, 3, 6, 5, 1, 7, 1, 8, 2. That clear? And yes, that's the right answer. That's the right answer. Sorry, there. Okay. All right, so let's try to do percentages really quickly so you guys can finish that packet out. So if I'm doing percentages, what the first thing I want to find is the total number of the cells. So find total number of cells. So what we do is we count number across and the number of cells vertically up and down, right? And multiply. So for this example, I'm going to try to just zoom in a little bit here. Oh, okay. So for this example, we're just going to do it very quickly. It's not an exact thing. 1, 2, 3. I'm kind of counting this row. 4, 5, 6, 7, 9, 10. I think that 25 are up and down. And what about across? 1, 10, 30. I think that 35 are across. So I'm going to multiply 25 by 35. And I get 875. So 875 is the total number of cells. All right, now to get percent, I'm going to take the number in the phase. Okay, so if that's metaphase, prophase, metaphase, anaphase, telophase, or interphase, I'm going to divide that by the total number. Then I'm going to multiply by 100. So for prophase, how many did we find in prophase? Didn't we find 1, 2, 3? Yes, 3 were in prophase. I'm going to write a P, P, and a P. Remember that prophase, metaphase, anaphase, telophase, that's what you're going to be tested on. So I'm going to take 3 divided by 875, and then I'm going to multiply that by 100. Okay? 3 divided by 875 times 100. So, why is this not 875 times 0.34%. So not even 1% are in prophase. Hopefully that's making sense. So let's do metaphase. So what did I find? Let's do a different color. So we said that metaphase 1, sorry, I know it's really pale, but we see 1 and 2. So we're going to do 2 divided by 875 times 100 equals 2 divided by 5 times 100, 0.23%. 0.23%. Okay, now we're going to do anaphase. Anaphase, we found, I'm just reminding you too, we just found 1. Okay, so we're going to do 1 divided by 875 times 100. 0.12%. And then we're going to do 4 telophase. Okay, and we're going to just find those really quick. We said that it was this one here, this one here. So we're going to do 2 divided by 875 times 100. 2 divided by 875 times 100. 0.23%. Okay, so where would you write these? So if you go to your packet, I know there's a lot of writing. You're going to write these down right here. Okay, so I'll just do that really quick so we can show you how to do interphase. So for the number of cells, we said it was 3, right? 2, 1, 2. For the percent, we said it was 0.34%, 0.23%, 0.12%. And I'm going to let you guys do whitefish blastula on your own, okay? 0.23%. So now interphase. Interphase is all that's left. Now we know we had 875 cells total. So we're just going to take 875 and we're going to subtract 3 plus 2 is 5, 5 plus 1 is 6, 6 plus 2 is 8. So we're going to subtract 8 because 867 cells would be all that would be left, right? So everything that's not in prophase, metaphase, anaphase, telophase is in interphase. So when we add all these up, we get 875. Does that make sense? So interphase cells is 867. The percent, you can just take 867 divided by 875 times 100. and we get 99.1%. On your lab packet, if your guys' percentages do not add up to close to 100%, you will not get the points here. Okay, so we will be kind of checking your work. So we've had students write down, I'm not even kidding, I've seen packets where they wrote 40%, and then they said, you know what, I think 30% sounds good. Then they're like, you know, I think that like 40% again would be good. And then they say, well, I think 70%. And it's like, well, you can't have things that don't add up to 100% because we're trying to say how many are on the slide based on percentages. Does that make sense to everybody? So make sure all of your percentages are adding up to 100. And you're going to count prophase, metaphase, anaphase, telophase first, and then interphase would be last.
I have a question. So we got the 867 because the number of cells added up to 8 and we subtract that from 875?
Yep.
Okay.
That's how many you had in the other phases. Very good. So everybody good with doing their, sorry, I'm supposed to do it on number 8. So everybody would be doing number 10 by themselves because you would be using this picture. Yes, so you use this picture and you're going to do the same process, but with this picture. So the one we did was for the onion root tip. You can use my answers if you wanted to, but for the number, this one, you would be doing that by yourself, okay? Again, just the percentages. I already showed you the tested part. The tested part is identifying which phases are in prophase, metaphase, anaphase, and telophase. Yes? Good. Any questions on this? Confusion, comments? So you should be set up for completing 90% of your metric packet, your mitosis packet, and your microscope packet this weekend, so that next week you go into lab ready to do your tissues packet. Good. Maybe you want to look at one more mitosis slide under the microscope. Maybe you want to look at one more paramecium, but you're not sitting in there still trying to start from scratch. Does that make sense? You're kind of just brushing up on a few areas in them. And if you guys need help or you forget what will be on the test from the packet, you can always message me. You guys are welcome to message me and say, will this be on the test? If I'm not allowed to answer something, which really that's not the case, but I'm just telling you, I'll just say, oh, I can't tell you. Like, you know, if you say, will this specific thing inside of the lecture be a question? No, I can't like say what's going to be a question on your test. But if you say, hey, is this part of the microscope or this part of this on the test? I can't remember if you said we're supposed to do XYZ on the test. That's fine. I mean, I want to help guide you as much as possible. Does that make sense? So if there's something you're confused on, just message me and I should be able to clarify. All right, so let's do the mitosis models. Does that sound good to everybody? Do these mitosis models. So these will be on your lab exam. We will be testing you on these on your lab exam. So remember, get out your key. You can pull it up on your computer if you're in the lab. And this key will tell you what you need to identify. So it's the same for every model. So for the cell model, you look at your cell key and that tells you what you need to know from the cell model, right? So you need to know these terms. So most of the keys will say you need to know their functions. And that's because the functions will be tested for you guys mostly on the lecture exam. The lab exam is not a lot of testing on the functions. Okay, so I'll just tell you it's some functions, but not a lot of functions tested on the lab exam. Mostly it's identification. So we're going to point to something. So for example, here, maybe you'll walk up and you'll just see one of these. They're not going to all be together. And it will be pointing to a certain structure with like a piece of blue tape in this example. And it'll say, what is the name of this structure. Does that make sense? So most of the lab exam is just identification. Okay, I'm going to go through and label these. And then at that part, once I'm done labeling mitosis, we'll stop the SI session. And then we'll open it up for questions. So any questions that anybody has on any of the homework, like we said, we'll do some more metric problems. And then basically once we're done, anybody who's ready to go can go, and anybody who wants to stay can stay. And I'll probably start with some metric questions at 11. Okay, so G1 is here. S, I need a different color. So, okay. This is basically like S/G2. This is the beginning of prophase. This is true prophase. This is metaphase. This is anaphase because they've begun pulling apart. Anaphase, they're continuing to pull apart. Telophase, they're really fully pulled apart. And then this is the end of telophase. We call this cytokinesis. Cytokinesis is when the cytoplasm itself is actually dividing. All right, so that's the phases. So we do get more specific than the phases on the test. We want you to identify the certain parts of the actual cell. So these purple lines, we call those the spindle fibers. I'm going to try to, if you guys can't see something, let me know, but I'm just going to try to write over stuff. So spindle fibers, again, are kind of those purple lines. You also have the pair of centrioles that's called a centrosome. So these two little orange structures, so it's two centrioles called a centrosome. So what we say is we say together they're called centrosome. When each of them is separate, it's a centriole. So just to make it clear, this would be one centriole. If I'm talking about them together, that's a centrosome. Okay. When